Hello guys, today I will be talking about the neural regulation of respiration. We shall study this topic under two subsections. The first one is the automatic control of respiration and the second one is the voluntary control of respiration. So moving on with the automatic control of respiration. The first thing that we come across is the pre-Botzinger complex, which is known as the pacemaker of respiration. It is located between the nucleus ambiguus and the lateral reticular nucleus of the medulla, and its main function is to maintain the respiratory rhythm. Now we will move on with the medullary centers that control respiration. The neurons that are present in the medulla are divided into two subgroups, dorsal respiratory group as well as the ventral respiratory group. The dorsal respiratory group is located in the dorsomedial medulla as well as the nucleus of tractus solitarius. It gives rise to upper motor neurons, which supplies the external intercostal muscles as well as the diaphragm. The discharge of dorsal respiratory group is in a ramp-like fashion, which means it initiates slowly, then increases for next few seconds and then terminates abruptly. And as it terminates, expiration occurs passively due to the recoil of the lungs and the diaphragm. The function of the DRG is to cause inspiration, and as the expiration occurs passively after it, therefore, in case of DRG, normal inspiration as well as expiration occurs. Now moving on with the VRG. It is located in the ventrolateral medulla. The neurons are again further divided into three subgroups, rostral, intermediate and the caudal. The rostral one stimulates the caudal one, and the function of the intermediate is to stimulate the accessory muscles involved in inspiration, and the function of the caudal group of neurons is to stimulate the accessory muscles involved in expiration. The VRG functions only in case of strenuous exercise and similar activities. Now moving on with the pontine centers. The first one is the apneustic center. It is located in the lower pons and it stimulates the dorsal respiratory group. Due to the stimulation of the DRG, the ramp-like discharge increases and therefore prolonging the inspiration, which is called as apneusis. The next one is the pneumotaxic center. It is located in the dorsolateral pons. It is formed by medial parabrachial and the collicular nuclei. It inhibits apneustic center. And once the apneustic center is inhibited, the discharge terminates earlier. As a result, shallow and rapid breathing occurs. As we know that pulmonary ventilation is equal to tidal volume into the respiratory rate, so to maintain the same pulmonary ventilation, and if the apneustic center is inhibited, then the tidal volume will decrease. However, in order to maintain the pulmonary ventilation with a decreased tidal volume, the respiratory rate has to be increased. Therefore, shallow and rapid breathing occurs. Now moving on with the role played by the vagal fibers. The vagal fibers arises from the stretch receptors present all over the lungs, and it inhibits the apneustic center when the tidal volume crosses 800 ml, and it alters the depth of breathing. Now moving on with an experiment that was performed in which different sections of brainstem was done, and its effect on breathing was noted with vagi intact and vagi cut. So moving on to the first section that was done at the level of the upper pons. With vagi intact, there was no change in normal inspiration as well as expiration, but with vagi cut, the breathing became shallower and rapid. Now moving on with the next section that was done at the level of mid pons. When this section was done, the apneustic center became out of control of pneumotaxic center, and with the vagi intact, the breathing was shallow and rapid, but with vagi cut, apneusis was seen. Now moving on to the third section that was done at the level of lower pons. When this section was done, the DRG as well as the expiratory center became out of control of the pneumotaxic as well as the apneustic center. As a result, in this case with vagi intact and vagi cut, irregular inspiration and expiration continued, but it was spontaneous. The next section was done at the level of lower medulla, and when this section was done, the upper motor neurons supplying the diaphragm as well as the external intercostal muscles involved in inspiration were removed. As a result, cessation of respiration was seen. Thank you.